let's study about inhibitory gene interaction. So what is inhibitory gene interaction? As the term implies, one gene gives rise to some sort of expression and that is inhibited by another gene. And these two genes are located in different loci. Okay, so the expression of one gene is suppressed or inhibited by another gene. I will explain with an example. In paddy, two kinds of leaf colors are seen, purple color and green color. The purple color is due to the presence of a capital P allele. That is, P gene is responsible for purple color. The dominant allele of P is needed for the purple color. But at the same time, when inhibitory gene, we can call it as I gene, if present in dominant condition along with capital P, the purple color is not produced. P gene is inhibited and the color becomes, or the color of the leaf is green in that condition. So, I will explain it further. Say the scalpel represents P gene. This P gene is responsible for purple color. But when this, say this brush stands for I gene. When this capital, this is a dominant gene. See, we have got another recessive gene is also, recessive allele is also there. So when this dominant allele of I is present, the expression of P is, capital P is inhibited there and only green color is produced. So in its absence, it is purple. In its presence, it is green in color. It has no expression of its own. If both the dominant alleles are absent, the plant is green in color. Okay, hope you understood. Now you may be thinking, so under what condition it becomes purple? Okay, so this is the condition when it becomes purple. So these two are small I. No dominant inhibitory gene present. Here it is capital P. So small I, capital P is a condition when the leaf color is purple. So here it is homozygous in condition, right? Now if it is heterozygous, then also it is purple in color because one dominant allele is enough for purple color. Here no dominant I gene is there to inhibit its expression also. Hope you understood. Okay. So this is the condition, double recessive homozygous condition. It is also green in color. Right? This is also green in color. Now let's see what will be the F1 individual when two homozygous green plants are crossed. So it is double recessive homozygous parent. Now let's see another parent which is also green. This one is green. This is also green. See what's the difference? This is double recessive. It is double dominant. Why it is green? Here dominant P is absent. So it is green in color. Here though dominant P is present, as capital I is present, inhibitory gene is present, so it is green in color. So when these two plants are crossed, what will be the F1 individual nature of the F1 individual? See, only one kind of gamete is formed from the first parent. What is that gamete? This is the gamete that can be produced from the first parent. Likewise, here also only one kind of gamete, that is this one. So this is a gamete produced from the second parent. This is a gamete produced from the first parent. So what will be the F1? This is the F1 genotype, that is capital I, small I, capital P, small p. Is it okay? So the F1 individual has got the genotype capital I, small I, capital P, small p. How many types of gametes can be produced from this? We have to see the F2 individuals and F2 ratio. So what are the different kinds of gametes produced from this? This I can fuse with either capital P or small p. the small I can fuse with either capital P or small p. Okay. So one gamete produced is capital I, capital P, capital I, small p, and the first one, second one, then small I, capital P, small I, small p. So 4 kinds of gametes can be produced from the F1 individual. Now let's see the checkerboard for the F2 individuals, and also the F2 ratio can be identified from that. Okay. So see, this is the Punnett square that shows F2 generation. I told you the different types of gametes which are formed from the F1 generation. So these 4 kinds of gametes were produced from the F1 generation, that is capital I, small I, capital P, small p. That was the genotype of F1 individual, and from that F1 individual 4 types of gametes were produced. And this Punnett square shows the different genotypes and phenotypes of all the 16 offspring which can be produced from this combination. You can see only 3 offspring are having purple leaves. Okay, here capital P is present in all these 3 cases. At the same time, capital I is absent. I had shown this using scalpel and brush. Hope you remember. Here it is double recessive and it is also green in color. So you can see the dihybrid Mendelian ratio 9:3:3:1 is now converted to 13:3 because of the special kind of inhibitory gene interaction. So here once again the inhibitory gene interaction means one gene, the expression of one gene is inhibited by another gene. Okay. So this is the ratio of phenotypic ratio of inhibitory gene interaction, that is 13:3. Hope you understood. Thank you.